Hello and welcome once again to the series of lectures on skeletal muscle tissue. Today, what I want to cover is a summary of what we have been talking about for a while now, which is a muscle contraction. So I'm going to use a series of images from the textbook to pull it all together into one final picture. And the picture that I'm going to start off with is on page 311 of your textbook, and it is very appropriately entitled Summary of Events of Contraction and Relaxation in the Skeletal Muscle Fiber. Excuse me. So we've been looking at various aspects of this entire picture. We've broken down, for example, the neuromuscular junction, and we looked at what was happening there. And then we looked at the sliding filament mechanism and all of that. So what we're doing today is we're putting all those pieces into one single puzzle and summarizing what that is all about. So to begin with, let's remind ourselves of exactly what the state of affairs is for the thick and thin filaments prior to a stimulation of the skeletal muscle by a motor neuron. And this picture will help us summarize that state of affairs. So as you look at this picture, what we're seeing is the state of a relaxed muscle. And the key thing here, there's a number of key things, and one of them is that the calcium is contained within the sarcoplasmic reticulum, and the actin binding site for the myosin heads is being covered up by tropomyosin. So there is no possibility that we can form cross bridges, and the calcium is being sequestered into this space here. So that's the state of affairs of a muscle cell when it's in a relaxed condition. Notice also the situation in terms of the relative positions of thick and thin filaments. So we have overlap of thick and thin filaments primarily in the region of where the heads are, and the center of the sarcomere is relatively clear of thin filaments. Okay? So that's the state of affairs prior to stimulation. But what we're now going to do is stimulate the muscle cell. And what's going to happen is an action potential will be sent down through the motor neuron to the synaptic end bulb, and that will cause the release of acetylcholine, which will then bind to the receptors. So acetylcholine binds to receptors in the motor end plate, ultimately triggering a muscle action potential. Okay, so we're binding, the receptors open, sodium flows in, we change the membrane potential, that is to say we change the charge across the membrane to become more positive. That triggers an action potential, which is then traveling along the membrane. And remember, that action potential is a very shallow stimulation. It doesn't go very deep into the cell. It stays on the surface along the edge of the membrane. So we have these T-tubules that dive down into the cell and bring that stimulus with it, that action potential, and then it reaches the series of structures, the voltage-gated calcium channels, and the calcium release channels. And here we're going to turn to another image to look at in more detail what's happening here. So what's going to happen is we're going to get the movement of the voltage-gated calcium channels away from the calcium release channels, and that will allow calcium to spill out from the sarcoplasmic reticulum into the general environment of the cell. And those molecules, calcium, will bind to troponin. That will cause tropomyosin to roll off of the binding site on actin, for the myosin heads, and we will bind now and create the cross bridges. And notice now what's happening. Because we form these cross bridges, and we're going to go through a cycle, which we're going to talk about in just a moment, there's going to be a sliding of the Z disc towards the center. That is to say, the thin filaments are going to be driven or sliding in the direction of the center of the M line, and the sarcomere is going to collapse. It's going to get compressed, and the entire myofibril will get compressed, and the cell itself will compress, and we'll have a muscle contraction. Okay, and so that's the state of affairs that we're seeing right here. That's seen right here. A muscle action potential traveling along a transverse tubule triggers a change in the voltage-gated calcium channels that causes the calcium release channels to open, allowing the release of calcium ions into the sarcoplasm. The calcium ions bind to troponin on the thin filament, exposing the myosin binding sites on actin. And then we get a contraction. The myosin heads bind to actin, undergo power stroke, release, and thin filaments are pulled towards the center of the sarcomere. So this gives us what we need in order for us to have this, the contraction cycle. So as you know, what's going to happen is we're going to take a molecule of ATP, we're going to hydrolyze that ATP molecule and transfer the energy into the myosin head. The ADP, adenosine diphosphate, and the phosphate group remain attached to that head. We have now an energized head, and as long as we have exposed binding sites, we can form the cross bridge. So the myosin head binds to actin, forming the cross bridge. Then we're going to release the phosphate group, and we're going to get the power stroke. So we're going to get this movement of the head from this position to that position. So it's going to physically move, and that energy to move that is coming from that reaction where we took ATP and converted it into ADP. That movement slides the thin filaments towards the center of the sarcomere. So the myosin head pivots, pulling the thin filament past the thick filament towards the center of the sarcomere. That gives us our power stroke. And then we're going to release ADP. We're going to bring an ATP. ATP will now bind, and the myosin heads will become disattached, and we'll start the cycle over again. So this cycle is repeated thousands and thousands of times per muscle contraction. It goes on and on and on. How long does it go on? As long as we have calcium in the environment and we have the exposed binding sites on actin, we're going to get this kind of interaction between the myosin heads and the thin filament, this cycle that we've just talked about here. Okay. So as you can see, coming back to this image, we're going to start from the position where there is quite a bit of space in the center of the sarcomere with no thin filaments. As this process goes, we're going to slide those filaments towards the center, thus shortening the cell. And that cycle we just talked about will repeat itself over and over again until we stop stimulating the cell with the neuron. So the neuron has to constantly send a signal to the muscle cell for this whole thing to happen. As soon as the signal from the motor neuron stops, it's going to interrupt the whole flow of things. So when the action potential stops, that is when the stimulation of the motor neuron stops, then we're going to take the acetylcholine and break it down. The receptors close. Sodium, so we have acetylcholine esterase will break down the acetylcholine, and we will no longer have open receptors. No more sodium will flow in. That will stop the action potential, and we will return to the blockage of the calcium release channel. And at that point, the ATP, the calcium ATPase pumps, and these are pumps that pump calcium into the sarcoplasmic reticulum. So calcium release channels close. Calcium ATPase pumps use ATP to restore low levels of calcium in the sarcoplasm. Okay? And when that happens, then tropomyosin slides back over the binding site for the myosin heads on actin, and we no longer can form cross bridges, so that cycle is stopped and the muscle relaxes. Okay. So that's the story. That's the complete story, putting everything together that we've just learned. And so this is a very short presentation just to summarize what we've done and take all those different pieces that we've been looking at and put them together in one complete story. So the next lecture, we're going to talk about how we produce tension in a muscle. And what I mean by that is how do we determine how much force is going to be applied by a particular muscle to do a particular job? Because what we've described in this process is an all or nothing situation. If binding sites are exposed, we're going to have that constant cycle of binding, release, power stroke, and so on. So if this is an all or nothing situation, how is it that we can control the amount of force that is going to be applied by a particular muscle to do a particular job? And that's going to be our next lecture. So I will see you in the next lecture, and I hope this was helpful in summarizing everything that we've been looking at in the last few lectures.